Compared to the kids he grew up with, Kirk Haan's research career has turned into more of a marathon than a sprint. He grew up on a farm near Lloydminster, and he wants to be a physician. Kirk Haan started his undergrad in 2016, and his residency will likely take him closer to 2033. That's because Kirk Haan is also working on his PhD in osmoregulation, the way our bodies balance salt and water. Kirk Haan is our guest today on Researchers Under the Scope. Hi, I'm your host, Jen Cannell, and today we're recording at the Health Sciences Building with our guest, Kirk Haan, who's not a doctor yet. He's midway through medical school, and Kirk is in the final year of his PhD, one that's looking at osmoregulation, the way our bodies balance water and salt. Kirk joins us now, and hello, thanks for joining us here on the podcast.
Thanks for having me, Jen. It's a pleasure to be here.
So, doing your PhD and your medical school at the same time, this doesn't seem like a whole lot of people do it. Why are you doing it this way?
That's a very good question, and it's one that I get asked a lot. Because at the University of Saskatchewan, there are currently 2 MD PhD students. We'll be the second and third people to ever do it.
Oh, so like one person has ever done this before?
Before us, yes, and that was like 10 or 15 years ago. So it's actually been really nice, and I've been extremely fortunate to be able to do it alongside another student, and we're going through the ranks of med school and grad school together, and we're both set to finish our PhDs this year, and so we'll end up finishing med school together as well.
And I mean, looking back at, okay, as you were growing up, you're from Saskatchewan here, or at least we're going to claim you on the Saskatchewan side of the border. Did you always sort of know, I'd like to go to school for years and years and years and be a scientist?
Not at all.
Okay.
When I started university, my plan was to get in and get out. I wanted to do pharmacy, and I was eligible to do the pharmacy before they changed it to a 6-year program from a 5-year program. But the summer before I started university, I worked for one of my brother's friends who's a pharmacist.
Oh.
And through talking with him and working in a pharmacy, we both kind of came to the conclusion that I shouldn't do pharmacy, just because I found that I wasn't getting enough direct interaction in the medical field, and it wasn't hands-on enough for what I wanted.
And when you say hands-on and what you knew you kind of wanted to do, like at what point in your life did you decide, yes, I do want to be hands-on about this? Would it be sort of right before you went to U of S?
Yeah, that's a tricky question. I've kind of worked with my hands my whole life, just kind of between rummaging around on a farm and always kind of building things to the sports that I played. Using my hands was a big part of just my day-to-day life.
Yeah.
And using them to help people in a direct way kind of came about at the start of university. And that's where I kind of developed that desire to pursue medicine.
And you could have just pursued medicine, as so many people do. At what point did you go, okay, osmoregulation is actually pretty cool? Can you take us through that research side of your career?
Absolutely. I started doing research after my second year university. I was fortunate enough to secure a summer student position. And from then, this was in 2018, I just stayed on in the lab. I did 2 summer projects. I did my honors degree, and then I also did a master's degree before I started med school. So I started med school already having a graduate degree, but every time I got into the lab during those early formative years, I started figuring things out and being able to ask these more complex questions. And under the supervision of my supervisor, he was a really good role model for teaching me how to ask the difficult questions and where to go to find the answers. And that just inspired me to keep going, which is why I've been in the lab for so long.
Do you get a lot of questions about, geez, you're on the long road, or not so much anymore?
Yeah, that still comes up quite often. My best friends are the best friends that I've had since elementary school. So they've seen this journey. We all came to the U of S and did undergrads together, but now they're homeowners, they're having kids, and my wife and I are renting a home, and we will be for the foreseeable future because we're both in this long-term thing. So there's a joke that goes around in our friend group that maybe when I finish school, their kids will be finishing school as well.
Yeah, you can supervise their theses.
So exactly.
Oh, geez. And you also made a point of staying, like your lab supervisor is Dr. Tom Fisher. And you made you could have gone to do different projects through the different summers. Why stay? Why stay at the one lab?
Part of it is that Dr. Fisher is an incredible supervisor, and I feel like I could work for him for another 5 to 10 years and I would still be learning from him. I still feel like I'm learning as much from him now after 7 years as I did in my first summer in the lab. So a main reason to stay is just having his guidance and his supervision. Also, on the research side, because I've been the only student in my lab for a long time, the research questions that we're answering take a little bit longer to answer. But after every year, we seem to just be on the cusp of another breakthrough in our field of research. So it's like a drug that keeps me coming back for more.
Okay, you just can't quit it. And it being osmoregulation, the way we balance water and salt, what is it about these processes that kind of grabbed you and held your attention this long?
I think it's a very niche field. And I've always been interested in things that are quite niche, but our lab maintains that it's really important to understand what's going on inside the cells from a pharmacology standpoint, from a physiology standpoint, and from a healthcare standpoint.
Where would you say you see a better understanding of osmoregulation kind of helping patients and helping people into the future?
Yeah, so osmoregulation is a crucial homeostatic process, and it's always active. Sometimes we're aware of it if we're thirsty or we're craving salt. Most of the time, we're not aware that this is going on in the background in our bodies, but it affects every system in our body. Salt and water are essential to everything that goes on in the human body. So as we get older, our bodies start to dry out. We can become more forgetful, and we have an increased prevalence of things like chronic disease that requires medical management. And the more drugs you take, the more likely you are to have a drug interaction that causes a perturbation in this system that kind of messes it all up. And so understanding how these processes work can help us figure out what drugs might interact with other drugs to mess up this system. And you can see this clinically because one of the most common things that you see in the emergency department are people that, regardless of what their primary costs are coming in for, have some kind of fluid salt imbalance that needs to be corrected. And a lot of the time, these people, if you give them like a normal saline solution and help them rehydrate, they feel so much better. So that right there is just a very low-hanging fruit to see that, okay, we really need to understand this system, because then we can maybe reduce the number of people that are coming into the emergency department with these issues and save the emergency department for actual emergencies where they're life-threatening. Because a lot of the people that are coming in for these things, they're not super life-threatening. They can be, but if you're properly hydrated or you understand what medications you should be taking or shouldn't be taking, that makes things a lot easier.
Tell me specifically about the cells you're looking at and the hormones that you're looking at. What are their names?
Right. So I look at MNCs, which are a specialized type of neuron in the hypothalamus, and they secrete the hormone vasopressin, which is involved in regulating the amount of water that gets excreted from the kidneys. And in simpler terms, it kind of controls how much you pee. Okay.
All right. So the control P cells, but what is the question that you're trying to answer right now in the lab?
Right now, I am trying to answer the question of how these cells work over long periods of time to keep a high degree of activity and a high amount of vasopressin release over long periods of disrupted salt and fluid balance.
And when we say long periods, are we talking like days, months, years?
We're talking in cells in a dish, anywhere from 90 minutes to 24 hours, but these can have implications that extend for as long as the salty stimulus is present. So, these processes could go on for, theoretically, for years.
For as long as the humans.
As long as you have an issue with your salt and fluid balance, these processes will be relevant.
And what are some of the disease states where you do run into issues? Like, I get dehydration. It is hot out. I haven't been drinking enough water. Or I just go through my day and I don't have my water bottle with me. What diseases would be involved in this?
There are so many diseases that result in or have a side effect of dysfunctional salt and fluid balance. So some really popular ones that are very easy to name would be things like Alzheimer's or Parkinson's or amyotrophic lateral sclerosis, which is ALS. A lot of neurodegenerative diseases, as one of the sequelae of those diseases is dysfunctional fluid balance. And basically any person that's living with chronic disease and is taking multiple medications is also at a very high risk of suffering from this imbalance in their salt and fluid regulation, because the more drugs a person takes, the more likely they are to have a drug interaction, which can put this system out of balance.
And that's what leads to some of these neurological conditions, or it's at least associated with them.
Yes, so I wouldn't say that it causes. If it did cause, then our research would probably be a bit more of a hot topic than it is. But certainly in those diseases, they affect other processes in the brain that help us remember to drink water. And they affect other processes in the brain that tell other parts of our body to either get rid of or hold on to water. And so that's where those imbalances come in. And with some of the stuff that we're looking at right now, there are processes that exist in other parts of the brain using the proteins that we're looking at. And so what we're thinking might be happening is that those disease processes might actually have a role in the cells that we look at, which isn't really known right now. And so the proteins that we're specifically looking at right now are called synaptotagmin 11 and endophyllin A1. And I know that that can be a mouthful, and you might think, what on earth are these proteins doing? But we're finding out that they might play a massive role in the long-term regulation of our body fluids.
So when you're actually in the lab and looking at them and at their processes, what are you asking them to do? How are you testing?
That's a really good question. So primarily I work with cells that are in a dish. So I'll take a brain out of a rat and I'll isolate some cells, and then I'll perform experiments on those cells. And then I stain them with a specific stain, like an antibody. That allows us to see where the proteins are located in the cells. And we can do different manipulations with things like inhibitors or specific viruses that either cause more expression or less expression of one of our proteins of interest. And that allows us to look more at what these proteins are actually doing inside the cells under different conditions. So synaptotagmin 11 is part of a complex of proteins that's involved in basically fusing vesicles inside the cell with the cell membrane. And we're trying to figure out exactly how this protein interacts with proteins around it to regulate that movement. And this comes kind of as another jumping point from the research that I did during my master's, which was to determine that there are vesicles that contain specific ion channels that move back and forth between inside the cell and the cell membrane to allow the cell to remain in a highly active state for long periods of time. For example, as long as what we would call an osmotic stimulus is present. So as long as a salty stimulus is present.
Where does the salty stimulus come from?
That can be from not drinking enough water. That can be from a drug interaction. That can be from eating a whole Costco-sized bag of salt and vinegar Miss Vickie's chips. It could be a variety of things, but essentially anything that would cause an imbalance in the ratio of fluid to salt in your body would activate these cells.
Once they're activated, what does that mean?
once they're activated, they release that hormone, vasopressin. So that hormone then goes to the kidneys, tells it to reabsorb more water, and then that kind of acts as a buffer to help restore that osmotic balance.
Kidneys, help me digest. I've got all these chips. I've got to get rid of them. Help. Yeah. Okay.
Yeah. I accidentally ate a whole bag of chips and now my body is flooded with salt. Help me regulate that. And that's what those cells do.
It makes sense. And that's also why I need to drink more water when I eat chips. This whole podcast, all I'm thinking about is, gee, I probably should drink more water. Am I drinking enough water? Can you drink too much water?
Absolutely you can. The old adage is the dose makes the poison. And so water is essential for all life, but you can drink too much water and then you'll dilute your blood, you'll dilute the salts, and then you'll have too little salt, which I would say is probably actually more dangerous than having too much salt, because if you don't have those specific salts like potassium and sodium and calcium, you need those for all of your cells to function. And so if you don't have enough of them in your fluids, cells aren't going to function. And that's how you can go into a coma. You risk death and other really, really serious diseases.
You're drowning everything out. Don't do that. Okay. Can you take me back a little bit to what you did prove for your master's, and then how that's led to all these more questions for you and for Tom?
Sure. So during my master's, we had a hypothesis that this ion channel called TRPV1 that's known to be involved in this osmoregulation process in MNCs, we hypothesized that part of this long-term response is the TRPV1 channels moving from inside the cell to the plasma membrane. And when that happens, you have an increased ability to sense the changes in the salt concentrations around you. And when that happens, you can get more activity from those MNCs, which leads to more vasopressin release, which leads to better regulation.
Right. I can go to the bathroom properly. My kidneys are happier with me.
Exactly. And so what we discovered was that this process does happen. And then we discovered some parameters by which it is regulated. And one of those parameters was that it was regulated by that complex of proteins. So from there, our launch point was, okay, let's look at the different complexes of proteins that exist. And we found synaptotegmin 11 as a potential candidate for a protein that could be regulating this process. And so that's been one of the main parts of what I've been looking at over the last 3 years of my PhD. Our hypothesis right now is that this protein is regulating this ion channel translocation. And what we've seen from some experiments that we've done is that when you reduce the function of this protein, the cells stop working properly. So that's a really good key indicator that, hey, if you don't have this protein,
things don't work.
The cells don't work. So that's where a big part of the working hypothesis has stemmed from.
What about endophyllin A1? That's the other protein you mentioned.
Correct. Yes. So endophyllin is another family of proteins that have a variety of functions that exist in different types of cells. And the reason why we started looking at endophyllin is because we looked into the literature more deeply in synaptotegmin. And we found that synaptotegmin 11 is associated with this protein called endophyllin A1 in other neuron types. So we wanted to see, does endophyllin A1 play any type of role in the system that we're looking at? Nobody has looked at that before. So we're the first people to actually be examining that in this system. And it was really interesting because synaptotegmin 11 and endophyllin A1 are actually implicated in diseases like Parkinson's and ALS. And these patients that have Parkinson's and ALS, they often have this dysfunctional osmoregulation. So we're starting to put pieces together that, we've wiped the dirt off, we found the piece. Now we're trying to put that puzzle together and figuring out how the pieces work together to get a better picture of what's going on in our system and how it could potentially relate to other disease states.
And once you're through with your PhD, once you've managed to, prove, yes, this is involved and here's how, then what? Where are you publishing this?
Our plan is to look at all of the data I've collected so far and figure out if we want to try and tell a few smaller stories, or if we want to put our research eggs into one basket and tell one massive story.
What will this mean for you? Because by my clock, you have to get back to medical school at some point soon.
I do, and this is the big issue that we're facing right now is how do we triage where we submit papers to and still make it so that I can write my PhD thesis, that I can defend it, and then I can go back to medical school on time. We're trying to figure that out, and if it ends up that I have to do some edits while I'm, on a rotation in my third or fourth year of medical school, then that's how it happens. But even when I'm technically finished in the Fisher lab, I will be a part of the Fisher lab as long as it's running.
Well, because you were still going to visit the lab even during first and second year med school, weren't you?
Yes, I got a bit of a head start on my PhD, not just because I am doing it in the same lab that I did my master's in, but during my first 2 years of med school, I was still working 20, sometimes 30 hours a week in the lab, just making sure that the experiments were going well, that I was getting on the microscope for a good amount of time. And we still had semi-regular lab meetings even throughout my first 2 years of medical school.
This sounds really heavy. Yeah, I would say you don't need like a backyard to take care of and pets and children and that whole part of life yet. But like, how does this work on a personal level for you? Like, how are you managing?
I have an incredible support network. I have a wife. I'm a happily married man, and she's been the best support network. And I have an incredible community of friends in Saskatoon as well. My wife actually just finished her PhD in April, and she just started medical school this past week. So we're both going to end up with MDs and PhDs. We took different paths to get there, but we've gotten to see each other quite a bit in the health sciences building throughout the duration of the time that we've been together. And that's been a huge motivator. And to have her be there right beside me, understanding what I'm going through because she's going through the same thing, makes life so much easier.
That's huge. And it also makes me wonder, like, where are you guys going to end up? Like, as you look at, okay, beyond this campus, as you look at fellowships, residencies, where you're going to be, there's a lot of planning and coordination is going to have to go into that.
Right. And right now, my big interest clinically is in ENT, or ear, nose, throat surgery. And sadly, there's not a program in Saskatchewan for it yet. So I think we will probably have to move away to do residency. But the plan is that as soon as we're finished residency, maybe a year or two fellowship to subspecialize, we try and come back to Saskatchewan. We both love it here, and we find that our community is here, and we'd love to be able to raise a family and contribute to the growing culture of medical research in Saskatchewan.
Well, and no matter what specialty you take, can you see yourself continuing to like get back into the lab and to put research questions together?
Yeah, my dream is to be a clinician scientist. Not just a clinician scientist, but a surgeon scientist. And in the ENT world, there aren't that many surgeon scientists in Canada. And so I would hope to be able to use what I know from the basic science world and blend it with surgery to really create a lab that works on benchtop to bedside type of research. So we get to be a part of, let's create this thing in a basic science lab, let's get it through clinical trials, and let's see if we can actually improve patient care.
That would be really amazing. I wish you well with it. And Kirk, thank you just for being here and for sharing this with us on Researchers Under the Scope. Absolutely.
It was a pleasure to be here, and thank you for listening to my rants and raves.
I like them. They're good. Kirk Haan is one of the very rare students at the University of Saskatchewan doing both his MD and his PhD at the same time. Dr. Thomas Fisher is supervising Haan's doctoral work on osmoregulation, and Kirk Haan is set to return for his third year of med school next summer. And by the time he's finished his residency, it will be about 2033, and that doesn't count any fellowships or postdoc work. And To me, this guy seems like he's in it for the long haul. You can find out more at medicine.usask.ca. Researchers Under the Scope is a presentation of the Office of the Vice Dean of Research at the U of S College of Medicine. We record and produce this podcast on Treaty 6 territory. We pay our respect to the First Nations and Métis ancestors of this place. We reaffirm our relationship with one another. And we'd like to thank you for listening, for being a part of the show. I'm Jen Cannell. I'm your host. Thanks for listening to Researchers Under the Scope.